Have you ever wondered why we can send a spacecraft perfectly to Mars, but we can't seem to predict the exact path of a hurricane or map the incredibly complex circuitry of the human brain? Well, welcome to this explainer. It turns out we might have actually been using the exact wrong kind of logic to tackle our most interesting problems.
Think about a broken mechanical clock for a second. If it stops working, you can just take it apart, find that one single broken gear, replace it, and boom, the clock works perfectly again. But you absolutely cannot divide a cloud in half to figure out why there's a drought, right? Clocks are linear. They're predictable. Clouds are messy, complex, and dynamic. Yet, for a really, really long time, science has essentially treated biology exactly like a clock.
This whole clock-like approach has a name: reductionism. And for about 500 years, it's pretty much been the absolute bedrock of modern science. It operates on this really simple, almost undeniable logic. If you can just understand the individual pieces, you'll naturally be able to explain the whole. Understand the atoms, then you understand the molecules, and eventually you figured out the entire human body. Just sum up the parts.
Now, the core assumption driving reductionism is this idea of 100% predictability, meaning if you know all the pieces and all the rules at the starting state, you can perfectly predict the mature end state. And what if you see some messy variation along the way? Well, under this paradigm, that variation is literally just dismissed as an annoying error in our instruments. It's just treated as mere noise, rather than, say, a meaningful feature of the whole system.
To see exactly how this clock-like logic got applied to biology, let's look at how we first mapped the visual cortex. Some incredible Nobel-winning research showed what looked like a perfectly reductive, step-by-step assembly line in the brain. Basically, specific neurons only respond to little dots of light. Then, those feed into the next layer to recognize straight lines, which then feed into another layer to recognize complex curves. Honestly, it looked like the ultimate victory for reductionism.
But if we take that linear logic and follow it all the way up to the top of the brain, we're forced to ask a pretty fascinating question. Is there a specific grandmother neuron? Because think about it. If the brain is just linearly stacking these recognizing neurons, eventually you'd have to reach a single, highly specific cell sitting at the very top of your visual cortex whose absolute only job in the world is to recognize your grandmother's face.
And incredibly enough, scientists actually thought they found exactly that. They discovered a single sparse coding neuron in a monkey that literally responded exclusively to pictures of Jennifer Aniston. And get this, it completely ignored pictures of Brad Pitt and Julia Roberts. No joke, it was a bizarre, absolutely stunning finding that, at the time, really seemed to validate this step-by-step, linear view of the brain. But here is where the entire paradigm spectacularly breaks down, and it falls apart for three main reasons. We lack the neurons, we lack the genes, and we just can't escape random chance. Let's start with the neurons. The simple truth is there just aren't enough of them in the human brain to process every single face, object, and concept using this linear math. If we really needed a unique neuron for every single thing we recognize, we would run out of brain space almost immediately.
We hit our second roadblock, which is a lack of genes when we look at bifurcating systems. These are the fractal branching networks that literally keep us alive, like our blood vessels and our lungs. These structures branch out, and then they branch again and again, creating these infinitely complex patterns. Now, a purely reductionist view claims we'd need a specific gene to act as a localized blueprint for every single one of those millions of branching points.
The problem: the math just simply doesn't work out. We have millions of these complex biological branches inside of us, but we only have roughly 20,000 genes. We literally run out of genetic pieces to code the human body like it's some sort of mechanical blueprint. So right there, the reductive model totally collapses under the sheer weight of our own biological complexity.
And finally, we have to introduce the wildly chaotic element of chance. Even when you have the ultimate identical starting state, like say, twin embryos, pure chance dictates that their very first cell split will actually unequally divide their mitochondria. So while a reductionist says an identical start must equal an identical finish, biology throws a curveball and introduces random, chaotic noise at the literal very first moment of life.
And this unpredictability applies to behavior too. Researcher Ivan Chase actually proved this beautifully with fish. He found that knowing the perfect one-on-one dominance between isolated fish gave absolutely zero predictability for how they would act in a group setting. Once they were all placed together in a social tank, random movements and chance encounters basically just created sheer chaos. The whole system ended up behaving entirely differently than just the sum of its isolated parts. All of this leads us to a pretty profound realization.
Biological systems are not linear, mechanical clocks that can just be taken apart piece by piece. They are complex, nonlinear clouds. Whether we're studying the brain, or genetics, or social behavior, we are essentially studying a cloud. You can't just take apart the gears in a vacuum and expect to magically know how the weather works.
Now, does this mean the last 500 years of science have just been a total waste? No, not at all. Reductionism is actually highly effective if you just want generalities, things like average vaccine efficacy or broad seasonal weather trends. If you just want to know that July is generally going to be warmer than January, reductionism is absolutely great. But if you want to predict the exact temperature on a Tuesday 3 years from now, it completely fails.
So, as we wrap up this explainer, I want to leave you with this final thought to really ponder. If the messy variability we see all across biology isn't just a measurement error that we need to somehow fix, what hidden chaotic rules is life actually following? Because when you start seeing variation not as a mistake, but as the fundamental nature of life itself, it completely changes how you understand the world. Thanks for diving into this with me today, and keep questioning the clockwork.